capillary fluid exchange, this is a humongously important concept because it is crucial for understanding how edema occurs. So fluid transfer across capillaries is an important equation to know by memory, and also conceptually, because so many diseases can disrupt the balance of these Starling forces. Starling forces referring to the hydrostatic pressure and osmotic pressure that exists within capillary fluid exchange mechanisms. Examiners absolutely love this equation because it tests your understanding of the different reasons for having fluid shifts, which is something that is important in the cardiology section, as you will see again when we reach heart failure. Let's define some terms before we start talking about how this section describes physiologic function. First, PC means the hydrostatic pressure in the capillary, and that is the actual difference in fluid pressure between the capillary lumen and the interstitium. So this difference in pressure. Fluid will always move from areas of high to low pressure. Hydrostatic pressure is always denoted by P, and in this situation the PC represents the intracapillary, hence the C, pressure, and the pi symbol represents the pressure due to oncotic or osmotic pull. So again, C is capillary, I is interstitial component or compartment. The osmotic pressure is determined by the ability of negatively charged plasma proteins. So if we're talking about inside the lumen, we're talking about negatively charged plasma proteins like albumin. Remember, albumin is very negative, that's why it binds calcium. So intracapillary C, you have plasma proteins attracting water because water is polar, and the positive pole of water will be very attracted to these plasma proteins. And in the case of the interstitium, there are a bunch of proteoglycans. And remember, we discussed proteoglycans, particularly glycosylated glycosaminoglycans when we talked about heart valves. So in this situation, the osmotic pressure in the interstitium will be denoted by pi with an I, because pi is for oncotic or osmotic, and I is interstitium. So the proteoglycans in the interstitial space are also very negative, and these are responsible for pulling water out of the capillary into the interstitial space. So again, let's review. Plasma proteins are responsible for the osmotic pull into the capillary, and that is denoted by pi, which is for osmotic, and C, which is for capillary. Proteoglycans are responsible for the pull from the capillary into the interstitial space, which is denoted by pi with an I, because pi is for osmotic and I is for interstitium. So what about the KF or the JV that is present in your first aid book? So this is just a constant. It is a constant value that is determined by the junctions between endothelial cells that make up that capillary barrier. You can only change this if you disrupt the integrity of these junctions. That's why it's usually a constant. If you disrupt the integrity of these junctions, then that means something really bad happened, like acute trauma, like a knife, straight through. You can actually do it with other ways. You can do it with toxins, infection, due to cytokine-mediated change of the endothelial cell junctions. You can do it with histamine, which causes opening of these junctions, makes them very wide, and particularly where you can have a very high yield concept is a burn, which can also change the dynamics of the endothelial cell junctions. In burns, you actually just completely obliterate all of the capillary, and you have no way to keep the fluid in the intracapillary space. That is why burn victims have to be aggressively rehydrated. Now JV is the product of multiplying the filtration constant, or capillary permeability, times the net filtration pressure. This just tells you the total flow from the hydrostatic pressure and the oncotic pressure difference, while also incorporating the consideration of endothelial cell junctions. So capillary permeability is the endothelial cell junction component, and then the net filtration pressure is just the difference between the hydrostatic pressure pushing fluid out of the capillary versus pushing fluid back in the capillary versus the osmotic pressure pulling water into the interstitium and pulling water back into the capillary. The equation components are important to consider because they will tell you if you're going to have net filtration in one way or the other, that is, into the interstitium or into the capillary. If you have a positive hydrostatic pressure difference, this means that you will drive fluid out of the capillary. On the contrary, if you have a positive oncotic pressure difference, this will drive fluid back into the capillary. Look at the small diagram on the left that is directly from your Step 1 book. Do you see how the capillary hydrostatic pressure on the arteriolar end, so this is the arterial end, and this is the venular end of the capillary. So as you can see here, the PC, representing the capillary hydrostatic pressure, is pushing fluid out of the capillary. And on the venular end of the capillary, we have the interstitial hydrostatic pressure pushing fluid back in to the capillary. Why is this happening? Well, this is because the initial resistance on the arteriolar end of the capillary is very high. It is much easier for fluid to flow out. Now by the time we get all the way to the venular end of the capillary, all of this fluid has flown out. So there will be a gradient that favors it to re-enter the capillary because there's less resistance, because we lost a lot of fluid initially on the arteriolar end of the capillary. The same thing goes for the oncotic pressure. On the arteriolar side, we're pushing fluid into the capillary because we're pushing fluid in to the interstitium, and that means that we're increasing the concentration of the plasma proteins in the capillary. They cannot leave the capillary. We're increasing the concentration because we're losing fluid that would dilute these out. Therefore, there will be a net flow of fluid on the arteriolar end due to this newly created osmotic gradient from the loss of fluid due to the high level of hydrostatic pressure within the capillary. Now when we get to the venular end, we've lost a bunch of fluid because we pushed it all out this way, and we're starting to push fluid back into the capillary. Therefore, we're increasing the concentration of interstitial proteoglycans, which I'll abbreviate PG, and again this will create a new gradient in which water will see that we're increasing the concentration of these interstitial proteoglycans, and now there's a gradient for fluid to leave the intracapillary lumen and go chase down these negatively charged proteoglycans in the interstitial space. Let's discuss some situations in which we would have edema and how these individual components of the equation reflect the physiology of what is actually happening. So let's say a patient has congestive heart failure on the left side. It doesn't matter right side, left side. The physiology behind what's happening to these capillaries will be essentially the same. But whenever you have left side heart failure, it eventually develops into right side heart failure due to the backup of pressure and volume. Do not worry. Do not worry, we will discuss the pathophysiology behind CHF later. Mainly, let's just consider a patient in which the right ventricle is extremely non-compliant because it has had to hypertrophy so much to pump against the high pressure that is backed up into the pulmonary vasculature because our patient had left side heart failure. Remember that the edema in the pulmonary vasculature, hence pulmonary edema, is caused by the backup of blood and pressure into the pulmonary vasculature, and this causes hypoxic vasoconstriction, which will put a lot of strain on that right ventricle because normally the pulmonary vasculature is a very low resistance circuit, so the right ventricle does not have to work that hard. But we have changed that due to our left side heart failure. Our right ventricle will get backed up and congested just like the left side, and we will have right-sided heart failure. And this means that we increase the pressure and volume in the right ventricle because it cannot forward perfuse into the pulmonary vasculature anymore. So that means that we're going to have a bunch of difficulties. We can't dump into the left side of the heart, we can't dump into the right side of the heart. So the veins returning to the right side of the heart will have a problem because remember, venous return is calculated by the venous pressure minus the right atrial pressure? Well, we've caused right ventricular hypertrophy that led to right-sided heart failure. We're going to increase the pressures in the right ventricle and the right atrium. When you substantially increase the pressure in the right atrium, you lose this gradient or difference between these, and you have no driving pressure to return blood to the right side of the heart. This means that we now start getting venous congestion. So blood will just be chilling inside these capillary lumen in the veins. So that means that blood will just be backed up and chilling inside the veins and capillaries that are trying to drain to the right side of the heart. Now, what do you think this does to the hydrostatic pressure in the capillary lumen of the veins trying to drain into the right side of the heart? It is going to drive this sky high. The capillary pressure will increase, and we will have net movement of fluid into the interstitial space. This is why heart failure patients usually have incredibly prominent pretibial edema and can have other fluid transudits such as ascites. So they give you some numerical values next to the variables of the equation that we just discussed. The main one you need to focus on that's very abnormal is that the interstitial oncotic pressure or pull is very elevated. The question asks you for the mechanism of action of the drug that you gave the patient for treatment. Board examiners love this kind of stuff. So when we were talking about it, the patient went to Zimbabwe and bathed in a river. You were probably thinking about a microbiology question that was about to come your way. Maybe something like African river blindness caused by Onchocerca volvulus. So let's not forget the African river blindness is caused by Onchocerca volvulus. Not a bad guess, but number one, the patient's not suffering from blindness, and there was nothing about a black fly or bite, which is how it is transmitted, because the black fly carries the Onchocerca volvulus. So we eliminate that question answer, that process in our heads. So we kept on reading, and we saw some capillary fluid exchange numbers. We saw that the interstitial oncotic pull was incredibly high. We have to be thinking about something that we don't normally have in that interstitial compartment causing an increase in oncotic pressure. Well, why do they give you these variables with an increased interstitial fluid colloid or oncotic/osmotic pressure? Well, the organism that you need to be thinking about here is Wuchereria bancrofti, which is a filarial nematode. Remember, it's transmitted by a mosquito, and the mosquito will allow this organism to enter into the bloodstream. When it gets into the bloodstream, it then enters the lymphatics. So it's not actually going to stay in these capillaries. It's going to go straight to the lymphatics, mainly in the inguinal nodes, and it will start to replicate. The high number of organisms will block the lymphatic drainage. So the lymphatic drainage drains our interstitium. So remember, it goes capillary, interstitium to lymphatics. When you block these lymphatics, these get very engorged, and it disrupts the cell barriers that compose the lymphatic vessels. When your lymphatics are chock full of these organisms, you're going to eventually have a disruption of the integrity of these junctions between cells that line your lymphatic vessels. That means that a bunch of proteins and cellular debris and possibly even some organisms can leak into the interstitium. So they leak out of the lymphatics because we destroy the integrity of our lymphatic vessels. And now you have a bunch of extra proteins and cell debris chilling in your interstitial compartment. Now, what does this do to the interstitial oncotic pressure? It will drive it super high. Now, which drug would you use to treat this? First of all, I'd already be impressed with myself if I could remember Wuchereria bancrofti, nevertheless say it. And how am I supposed to remember which drug to treat it with, and then I have to remember the mechanism? Yep, unfortunately, board examiners will want you to dig that deep into your knowledge bank. The drug to treat Wuchereria bancrofti is diethylcarbamazine. It inhibits arachidonic acid metabolism in the organism. Remember, the arachidonic acid cascade is very important. That is how you make your prostaglandins. So this blocks the arachidonic acid pathway, which means you don't make any prostaglandins, and without any prostaglandins, you do not get any contraction of smooth muscle. Guess which organisms rely on contraction of smooth muscle for movement. Filarial nematodes like Wuchereria bancrofti rely heavily on smooth muscle contraction for locomotion. You paralyze these suckers and they're just going to die because they can't eat, they can't do anything. So that is how diethylcarbamazine will limit these organisms from moving, and therefore they will die.